Welcome to session 35. This is a continued lecture of mechanism of enzyme action. So this is mechanism of enzyme action 2. So here we will discuss one example how actually the enzymes break down the substrate into the product. By taking the mechanism of action of chymotrypsin, we will see here the structure of the substrate. We will see how we'll see the residues of the active site that are present in the enzyme chymotrypsin, and these residues, where they act and what are the products. All these things we will look into this session. So during digestion, dietary proteins must be broken down into small peptides by proteases. So chymotrypsin is one of several proteases that cuts peptides at specific location on the peptide backbone. This protease, chymotrypsin, is able to cleave the peptide bond adjacent to aromatic amino acids. So this we have to remember. So this is an aromatic amino acid, and this is our C double bond ONH, which is a peptide bond. So chymotrypsin acts at the residues, acts at the peptide bond near to the aromatic residues. And this is our amino terminal, and this is our carboxyl terminal, and so this is our peptide bond. Yeah, chymotrypsin uses most of the enzymatic mechanisms. So this is A chain, B chain, and C chain of the enzyme, and these are the residues. We can see here glycine 193 and serine 195, histidine 57, and aspartic acid 102. So these are the major amino acids that are present in the active site of the active site pocket of the enzyme. And this is we can see here the substrate is a small molecule that is present in the cleft or in the pocket of the active site of chymotrypsin. So this is a chymotrypsin free enzyme, and this is the hydrophobic pocket. When substrate binds, the side chain of the residue adjacent to the peptide bond to be cleaved, it accommodates in a hydrophobic pocket on the enzyme, positioning the peptide bond for attack. So this is the hydrophobic pocket of the enzyme, and the substrate molecule, which is a polypeptide, it gets accommodate itself into the hydrophobic pocket of the enzyme, positioning the peptide bond for the attack. So step 2, which is a nucleophilic attack. So here, after an enzyme substrate complex is formed, there is an interaction of the serine 195 and histidine 57, generates a strong nucleophilic alkoxide ion on serine 195. The ion attacks the peptide carbonyl group, forming a tetrahedral acyl enzyme. So this is accompanied by formation of a short-lived negative charge on the carbonyl oxygen of the substrate, which is stabilized by hydrogen bonding in the oxygen hole. So this is where we can see this. The third step is the substrate cleavage, and here the short-lived intermediate acylation, there is instability of the negative charge on the substrate carbonyl oxygen, leads to the collapse of the tetrahedral intermediate and reformation of a double bond with carbon displaces the bond between carbon and the amino group of the peptide linkage, breaking the peptide bond, and the amino acids amino living group is protonated by His 57, facilitating its displacement. So here there is a breakage of the peptide bond. The step 4, water comes in. And incoming water molecule is deprotonated by general base catalysis generating a strong nucleophilic hydroxide ion. Attack of hydroxide on the ester linkage of the acyl enzyme generates a second tetrahedral intermediate with oxygen in the oxanion hole, again taking on a negative charge. Then step 5, which is a very short-lived reaction. There is a collapse of the tetrahedral intermediate, forms the second product, a carboxylate anion, and displaces serine 195. So we can see here. So this is the last step, break off from the enzyme. The enzyme product 2 complex collapse of the tetrahedral intermediate forms the second product, a carboxylate anion, and displaces serine 195. It displaces the serine 195 and the product dissociates. So this is the product 2 where we can see there is a break in the peptide bond and the product is being released from the it dissociates from the active site and the free enzyme is regenerated. So this is about the mechanism of the action of chymotrypsin, where we have seen how amino acid, the how the polypeptide chain accommodates itself into the hydrophobic pocket of the chymotrypsin enzyme, and namely 4 amino acid residues. They play a major role. In breaking the peptide bond, and the role of water molecule, how it cleaves the peptide bond, and how the products are being released. So this we have seen in this session. Thank you one and all.